Hello, good afternoon, students. Can you hear me?
Yes, ma'am.
Yes, ma'am.
So today we're going to have a revision class on instruments and glassware. So the first one is a test tube. You all are familiar with it. It is used to hold, mix or heat small quantities of chemicals. So this is basically from a practical point of view. Whenever we give you any glassware, we want you to basically identify the glassware as well as you have to mention the use of the glassware. Other one is the centrifuge tube. You're familiar with it. It is a calibrated test tube which has got a conical tip at the bottom. And this is one centrifuge tube. Another type of centrifuge tube is something like this, which is the smaller one.
Yes,
And
ma'am.
this one we are going to put into this machine called as the centrifuge machine. So here they have got different slots for inserting the centrifuge tube, and it uses the principle of sedimentation principle. Okay, so whenever we put some acceleration into it, the denser substances and particles will move into the radial direction. At the same time, objects which are less dense, they get displaced and they will move to the center. So this is the basic principle of a centrifuge machine. So if you get this machine for identification, then make sure that you can identify it properly. There should be no confusion in the exam because these are all carrying marks. At least full marks, one full marks you can score by identifying the instrument as well as mentioning the use of the instrument. Okay, moving on. This is a cuvette, and this one, in which instrument is it used? Anybody can tell me?
Spectrophotometer.
Yes, photometry, yes.
Spectrophotometer.
Okay, basically for you all, you're dealing only with which machine? Colorimetry, okay. So this cuvette is the one which you're going to see in your colorimeter. So kindly identify and mention the correct spelling, because in the last exam, we have noticed that instead of colorimeter, many of you are writing calorimeter, okay, which is a completely different instrument. Is it clear? So this is a colorimeter, and the test tube which I showed you is the cuvette. So you should be able to identify between a normal test tube and a cuvette which is smaller and it is uniform diameter all round. Okay. So can anybody identify this instrument? You can see the plate here. Does it ring a bell for anybody? This is a ELISA, okay, ELISA, ELISA. I think a class was taken in ELISA by one of the teachers. So this is a ELISA machine and this is the ELISA plate, okay. So we're moving on to flask, okay. The first one is the conical flask, or called as Erlenmeyer flask. And this one, it can come in different sizes. The next flask is a round bottom flask which is used for saponification test, okay. So you have to make sure that you can identify the round bottom flask from a flat bottom round flask. You can see the difference here. Below it is flat bottomed, so it can stand on the surface, while the previous one, it is completely round bottom flask and it cannot stand on its own. We have to balance it on the other end. Okay, so in the exam, whenever we give you a flat bottom round flask, make sure that you identify it properly, because in the final exam we are going to be very particular that you can identify the glassware with the correct spelling. In the last exam, many of you have identified this as just as a round flask, not as a flat bottom round flask. So the same mistake should not be made. Can you make out the difference between both the flasks, between the flat bottom round flask and the round flask?
Yes, ma'am.
Yes, ma'am.
Okay, moving on. These are the beakers. So this can come from varying capacities from 5 to 5000 ml, and it is used mainly for preparing solutions, and it can also be used for heating the various solutions. Moving on. We have the volumetric flask. What happened is that in one of the exams, when we gave this volumetric flask, many of you have identified it as another glassware. So make sure that this is a pear-shaped flask, okay, and it has got a stopper at the top, okay, it has got a lid at the top. So please identify this as a volumetric flask. And it is for storing a particular volume of the chemical that we need. So this is the measuring glass cylinder. This one also comes in varying sizes. So if you see the smaller version of this in the exam, don't get confused. It is still a measuring glass cylinder, okay? So this is very easy. This is the glass funnel. Can anybody tell me the uses of a glass funnel?
Filter or transfer
Yes.
liquids.
Yeah, so filtering, transferring liquids. Anything else? In which experiment have you used this in your practical? Where you put the filter paper? Which test?
Biosalts.
Yes, yes,
Experiments.
yes. Correct. And this is a reagent bottle. It can come as a transparent, in the transparent reagent bottle, and this is the colored reagent bottle. So what is the advantage of this amber colored reagent bottle is that it can store reagents which are flammable, and even for keeping light sensitive chemicals like chloroform, ether and all. So whenever we give you either of this, you have to identify it as either a transparent reagent bottle or a amber colored reagent bottle. Don't just write reagent bottle, okay? Now coming to the various kind of pipettes. Pipettes are used for dispensing controlled quantities of liquids. So the volumetric or the bulb pipette, I think you all are familiar with it. It has got a bulb, even in I think physiology you're using it. And the main confusion happens in case of your graduated pipette, which is of two types. Okay, one is the serological pipette and one is the Moore pipette. And it is used to transfer known amount of liquid from one container to another. So can anybody identify here which one is this? This is the, for example, this is A and this is B. Can you identify which one is serological pipette from these two?
B. B.
Very good. So why do you say this is a serological pipette? How do you differentiate between them?
The markings are going till down to the end in case of a serological pipette.
Okay, good. So which one is the blowout pipette? Other name?
The Morse pipette A.
Can you hear me? So basically you have to be able to identify these either of these pipettes properly in the exam, okay? Don't just mention graduated pipette, you have to mention whether it is a serological or a Moore pipette, okay? This is the micropipette which you are handling. Can you tell me one experiment when you are handling micropipette? Any experiment that you remember where you have handled a micropipette? All of you have handled a micropipette?
Yes, ma'am.
You still remember how to handle it, no?
Yes, ma'am.
Okay. So any experiment that you can think of where you have handled it? Is it in case of
Urine analysis.
urine analysis? Yes. Abnormal constituents of urine? Yes. Anything else? Have you used it for any kind of analysis, estimation?
Yes, ma'am. Yes, ma'am. Protein estimation.
In all the estimations, you have used a micropipette. All right. And then the tips which is there, we just call as the micro tips. Okay, so sometimes if the micro tips are given and then somebody asks you what it is, you should be able to identify the micro tips. Okay. All right. And this is the burette. All of you are familiar with it. You have used it in your school days, in your high school for experimentation. This is used for titration and to dispense the corrosive reagents. So please make sure that you write the spelling as burette. In one of your papers, I think some of you have written it as a biurate reaction. So this is a burette, okay? So I think any doubts are there regarding any of the glassware's instruments? You'll be able to identify it.
Yes, ma'am.
Okay. Last but not the least, this is the urinometer. So it measures the specific gravity of urine at a standard temperature of 20 degrees Celsius. Does anybody remember what is the specific gravity of urine?
1.012 to 1.024.
Actually 1.0052, 1.030, okay. So you should be able, whenever any kind of glassware comes, you can, the kind of questions that you can expect is, when I give you this, for example, urinometer, I'll be like, identify the instrument. I can ask you the use of the instrument, or I can even directly ask you what is the specific gravity of urine. Okay, that time you should be able to at least answer the specific gravity of urine. Okay. Same thing for any other instrument. Okay, for example, we give you a stand which has got different color reactions. Okay, for example, for Benedict's test, what will be the color of the, if it's positive, what will be the color of the precipitate in the test tube?
Red.
Okay, it can range
Brown.
anywhere from green, orange.
Green, yellow, red,
Yes, yes.
blue.
So if we give you any kind of test tube with the Benedict's test, now you should be able to identify the test looking at the test tube. Okay. For example, if I give you what do you call it, a test tube for which a purple color ring is found at the junction of the two liquids, what test is it?
Ketone bodies.
Ketone bodies, presence of the ketone bodies.
Yes. So what is the name of the test?
Rothera's test.
Rothera's test, right? Same thing. Even if I give you any test with, for your bile salts, what is the test that we do?
Faucet test.
Okay, then for your bile, for the blood and urine, what is the name of the test? Anybody remembers? At least the name of the test? It starts with a B. Anybody? Blood and urine. Which test do we do? What is the name of the test? It's benzidine test, okay? So in that way, whenever we give you any kind of test tube, and we tell you that this is for doing a urine abnormality test, you should be able to identify the name of the test by looking at it, or at least whenever we ask you for this, for ketone bodies, what test is done, you should be able to mention that Rothera's test is done. What test do we do for proteins in urine?
Heat coagulation test.
Heat coagulation test, very good. Anything else you can think of? Nitric acid test is also done, no? In the same way, for identification, if we give you any kind of reagent, for example, we give you sulfur powder, okay? And then we ask you, what test do we do for urine abnormality? What will you do? What test will you do?
Haze.
Presence of
Haze.
bile
Has a sulfur test.
So these are like the kind of questions that we can ask you. That time there should not be any, there should not be any doubt in your mind what to answer. Okay? In the same way, if I give you some glassware, like I give you this water bath, okay? So what test have you used this water bath for?
Urine
Sorry?
analysis.
Which test? Anybody remembers? Is it done for glucose estimation? So your homework is to go back and find out whether you have used this water bath in any of the biochemistry tests which has been done so far. Okay? So when you get a water bath picture, please identify it as a water bath. Okay? Okay, now vacuotainers. You're all familiar with vacuotainers. You have been to the blood collection room. You have seen how the sample was collected, how to deal with the patient, what steps to do, right? I hope you all still remember it. Do you still remember it?
Ma'am.
Yes?
Yes,
Okay,
ma'am.
so these are the different vacuotainers. So whenever we give you any of the vacuotainer for identification, you should be able to identify which vacuotainer is used for which test. So next table I'm going to show you. Okay. So if you're going to get the red color vacuotainer and then it is asked what is it used for, you have to say that it is used for doing biochemistry tests, like for doing viral studies, for blood bank testing. If you're going to give you a purple color, what is it used for? It is used for checking your HbA1c. In case of, we are going to use it for checking the hemoglobin percentage, the CBC, WBC count, all of these, okay? Similarly, for glucose, we are using a gray color here. gray vacutainer, and then for your blue color vacutainer it is used for doing coagulation tests like your prothrombin time, EPTT tests, all of these. Okay, so whenever we give you any vacutainer, you should be able to identify it. By now you're familiar with all the vacutainers and you should be able to know which vacutainer is used for doing which kind of test. Okay, any doubts so far? So you all are like, it's a revision for you for the glassware and instruments. If anything is given, and since it is very scoring, I think I've already told you in the beginning of the year, you can score, 5 out of 5 in case of your glassware and the users. Okay, so just brush up on the users, brush up on all the glassware, the instruments, go back. If you have any doubt, then you can contact me, okay? And then find out which test is used for water bath is used for which of your experiments which you have already done in your class. Any doubts you have regarding today's class?
No, ma'am.
No, ma'am.
Okay, I hope you all are safe. I hope you all are studying also, staying at home.
Yes, ma'am.
Okay then, chalo, take care then. That's all for today's class. Thank you.
Thank you, ma'am.